Continuing our exploration of heredity and genetics, this is going to be Unit 6, Part 2, Traits and Inheritance.
Mendel's principles. Mendel believed that there must be two sets of instructions for each characteristic. This was his final conclusion after all of his work with peas. True breeders would always pass on the same set of instructions to the first filial generation of offspring. Okay. And if you cross two true breeders with different traits for the same characteristic, the F1 generation would carry instructions for both the dominant and the recessive traits, having received these instructions from each parent, because that's the only thing that each parent could give. One parent could only give the instructions for the dominant trait, and the other parent could only give the instructions for the recessive trait. Okay, so in walks Walter Sutton about, 40 years later. He's an American scientist who in 1903 examined grasshopper nuclei, and he actually saw chromosomes pairing up and separating during meiosis. A year later, he discovered that those chromosomes actually contained information, genes. So basically his finding together with Mendel's findings really gave a very clear look as to what was going on. He found Mendel's units of inheritance. Okay, so let's look at what we now know. Okay, scientists now call the individual units that determine an organism's traits genes. A gene is a segment of DNA located on a chromosome that carries hereditary instructions from parents to offspring. So it's a set of instructions located in a particular place on a chromosome. There can be different instructions possible for each gene, and we call these different sets of instructions alleles. An organism typically receives only one allele from each parent. And you can sort of see that in this drawing over here on the right, that the DNA has the information on the left side you see a parent that has a dominant allele, on the right side you see a parent that has a recessive allele on the DNA, and they get separated out during meiosis so that you end up getting different combinations of things in the sex cells. If an organism inherits different alleles for a trait, one allele may be dominant over the other. We know that from Mendel. The alleles of a gene separate from each other when sex cells are formed during meiosis. And there it is in a nutshell. Alleles and meiosis. Okay, so how this works. Homologous pairs of chromosomes separate during meiosis. Since alleles of a gene are found in matching locations on homologous pairs of chromosomes, they also separate during meiosis. Okay, so if you look at the picture, we have just one little homologous pair here. They copy in the S phase of interphase, and then when they enter meiosis, the homologous pairs separate during meiosis 1, that first division of meiosis, and then the copies separate during meiosis 2, that second division of meiosis. And so each sex cell has just one allele type. All right, let's go back over genotype versus phenotype to make sure we're okay with that. How an organism physically looks is called their phenotype. Pheno, physical looks, right? And so that would constitute, in the example of the picture here, purple or white. Their genotype represents exactly what the paired alleles in the genes are, or the entire genetic makeup of the organism. So genotype relates to genes and alleles. So examples of genotype on the right side of this picture would be big P, little p, big P, little p, little p. You get the picture. Okay, there's another term we can use for genotypes, and these are homozygous and heterozygous. Because scientists traditionally represent dominant alleles with capital letters and recessive alleles with lowercase letters, genotypes that consist of two of the same allele for a particular trait, either big R, or little r, are considered to be homozygous for the trait. Homo is a root that means same, okay? So homozygous if you have the same allele on both of your alleles, okay? And these, by the way, were Mendel's true breeders, okay? They only had the same allele on both of their chromosomes. Genotypes that consist of two different alleles, like big R, little r, for a particular trait, are considered to be heterozygous. Hetero means different or other, all right? And these would be Mendel's F1 generation, because the true breeders would each give either the dominant allele or the other, and then the other true breeder would give the recessive allele, and together they would form a heterozygous genotype, big R, little r. Here's another picture of that. So here we got our true breeders, okay, over here on the left. And you've got them, you know, their alleles separating out during meiosis. So each parent, the big P, can only give big P alleles to their sex cells, and the little p can only give little p alleles to the sex cells. And then when those combine, your only possible combination is, sorry, that's not working. There we go, is big P, little p. A heterozygous individual. Okay, so let's look at a different example, a sort of fun example. You might recognize our little friends here. Say we have a true breeding red-eyed alien, and we'll give it the allele big R for the red eyes. So if it's a true breeder, it's going to have two alleles that are exactly the same. They'll be homozygous, so he'll be big R, which is what you see here, okay? And then we have the other true breeder, which is the recessive trait. It's a true breeding yellow-eyed alien. And because this is a dominant recessive situation, we're going to give it the alleles little r because it's recessive to the big R red. All right, so its genotype is going to be little r, little r. When we cross those two together, the big R parent can only give a big R to the baby. The little r parent can only give a little r to the baby. So all babies are going to be heterozygous, big R, little r, and carry the information for both of the traits. But because red is dominant over yellow, it will be red. So here's what that looks like if we do a Punnett square. All right, a Punnett square is something that allows you to show all possible resulting offspring, both the genotypes and the phenotypes, from a particular cross of parents. It doesn't tell you the babies you have exactly. It tells you the possible babies you could have. Okay, it's important to recognize that. These are just possible offspring. All right, so now we're going to do the F1 Self-Pollination Cross, so to speak, okay, or an example like that. So if we let two individuals that were both heterozygous for the big R, little r trait of red, okay, to mate, one parent can give either a big R or a little r, okay, and the other parent can do the same, can either give a big R or a little r. So if this parent here, if the big R went to the baby and the big R from this parent went to the baby, then the possible offspring would be big R, a homozygous red. If the big R met up with the little r from the other parent, then the possible genotype would be big R, little r, still red. Okay. If the little r from the first parent and the big R from the second parent met up, then you'd end up with big R, little r again, and that would also be red. But if the little r from the one parent met up with the little r from the second parent, then you would have a possible yellow-eyed baby. And if you count the ratio here, the ratio is 3 possible reds to 1 possible yellow. And there's Mendel's magic ratio. So you can see that you get a 3 out of 4 chance, or a 75% possibility of getting the dominant trait and a 25% possibility of getting the recessive trait. and you get that ratio of 3 to 1. All right, the only way that you phenotypically, the only way that you physically will see yellow eyes is if you get both recessive traits from each of the parents. Okay, that's the only way it appears. Okay, so you actually had 3 phenotypes that were red in that possible offspring example, and 1 phenotype that was yellow, although 2 of the phenotypes for the red were the same. I mean, it is basically you either have a big R or a big R little r as your possible phenotypes for red, and only the little r for yellow. Okay, so you can see that here, all right? Only 2 phenotypes, either the big R or the big R little r are your phenotypes for red, but only the little r will be your yellow, okay? This big R is still the same phenotype as that one. All right, so in Mendel's example, if we had a true breeding purple and a true breeding white, their first generation would all be big P, little p, because that's all they could be. All right, and we can now talk about a little bit to make sure you understand. I'm sort of talking around it already, but probability is the mathematical chance that an event will occur. It can either be expressed as a fraction or a percentage. Generally speaking, for me, if I ask for probability, I'm going to want you to express it as a percentage, okay? So if we do Mendel's F1 self-pollination, making the F2 generation, and we end up with that 3 to 1 ratio, all right, we have 3/4 are purple and 1/4 are white, which translates to being 75% purple and 25% white. So those are the probabilities of getting each of those phenotypes, all right, 75% purple, 25% white. The guy who invented Punnett squares, by the way, is a guy called Reginald Crendell Punnett. This little website allows you to click and drag and practice some Punnett squares. You may want to check it out. Since we're not doing it right this second, we may get to it. Yeah, I probably will get to it in class. I just don't know if it'll be this first day or the second day that we do this. All right, here's a practice problem. In apples, red color, big R, is dominant over yellow color, little r. If you cross a heterozygous red apple and one homozygous recessive yellow apple, and show the genotypes and the phenotype ratio of the offspring. Okay, the hardest part about genetics, you guys, is the vocabulary. Once you get your head around what exactly each part is asking and what it means, the rest of it comes pretty easily. Okay, so first off that we're going to do is we're going to talk about, okay, what is a heterozygous red apple? Okay, heterozygous again means that the alleles are going to be different. So that means that its genotype is going to be big R, little r. Okay? Then we have this homozygous recessive yellow apple. Okay, homozygous means the alleles will be the same, but it's telling you that it's going to be the recessive allele. All right, so the recessive allele is this one. So its genotype is going to be little r, little r. Okay, so we're crossing a big R, little r with a little r, and it's asking what are the genotypic and phenotypic ratios of the offspring. Okay, so we're going to need to do a Punnett square. So let's go on to the next slide, and I can do that for you. All right, so again, we have big R crossed with little r. Okay, so I'm going to make my little Punnett square. All right, if this parent is giving alleles, it can either give a big R or a little r to the offspring. However, if this parent can only give little r's, right? Okay, so if this allele and this allele meet up, we're going to get a big R, little r baby. If this big R, this is just like a multiplication table, okay? Basically, you just bring it over and you cross them together and you see what the combinations are. Okay, again, big R, little r. If this little r means up with this little r, it's going to be little r, And actually this little r and this little r also will make little r. Okay, so first off, genotypes. We have two possible genotypes. It's a 1-to-1 ratio of genotypes, which are big R, little r to little r. Okay? The phenotypes is also the same. It's also a 1-to-1 because these are two different phenotypes. Okay, 1-to-1 ratio phenotypes. And the phenotypes are going to be what they actually look like. And big R, because big R, little r, because big R is dominant, is going to be red. And then the little r will be yellow. Okay? That's all there is to it. Okay? The vocabulary sounds crazy, but you know, the actual doing of it is not that bad. Okay? So let's try another one. All right, tongue rolling. Tongue rolling, big T, is dominant over non-tongue rolling, little t. A woman who cannot roll her tongue is married to a man who is a tongue roller and is heterozygote for the trait. What is the chance that their first child will be able to roll her tongue? Okay, so again, we have to deal with what's being asked here. So a woman who cannot roll her tongue. Okay, tongue rolling is dominant. Okay, the non-tongue rolling is recessive. The only way you can get a non-tongue roller is if you have both alleles, if you're homozygous for that recessive trait. Because if you're heterozygous and have the big T, then you're going to be able to roll your tongue, okay, because it's dominant over the non-rollers. All right, so a woman who cannot roll her tongue is going to be little t, okay? It says she's married to a man who is a tongue roller but is heterozygous for the trait, All right, so again, heterozygote means that you have one of each allele. So he's going to be big T, little t. Okay, so this is the cross. And this is the same cross that we did a while ago on the other page. All right, so you have like, you know, one parent can give either big T or little t, this parent, and this parent can give either a little t or little t. All right, so that's a little one. All right, so possible offspring are, if we bring the big T down and the little t's over, are these. And again, we have a 1-to-1 heterozygous to homozygous recessive. All right, so if 2 out of the 4 possibilities are going to be able to roll their tongue, that translates as a half, right? Which is equal to 50%. That's the chance that their first child will be able to roll their tongue. The chance that they'll not be able to roll their tongue is the other 50%. Okay, it's also 2 out of 4. Okay, because these are the big T, big little t's. These guys can roll their tongue. These guys will not be able to roll their tongue. Okay? All right. Ooh, that's it. That's all we're going to do. We're going to do more practice with these at a later date, but I'm hoping you're getting a sense of what's happening with all of this. What we're going to do is start work on this in this lab 11B, which is on crazy traits, and it talks about what role does chance play in an organism's heredity. So you're going to need to open up OneNote, copy the lab into your personal folder, and start on it. You are going to be working with a partner. I'll let you know who your partners are. Both of you will need to record your answers in your own personal lab as you go. All of the equipment is on the tables. Please make sure that if you're taking equipment from a particular box, that the equipment ends up back in that particular box. Don't mix and match, because otherwise I'll end up with incomplete sets all over the place. Okay? All right, and that's it. Thanks, guys.